want to start the talk by thanking all of you coming, my friends, colleagues, trainees, and the Radcliffe staff. It has been a transformative year for me. And as you can see from the rest of my talk, although this project has started as a collaboration with Ari Samuel's group as well as Jeff Lickman's group more than a year ago, more than a year before I started Radcliffe a year ago, the opportunity I've been given in the last year has really allowed me to move the project forward, and exciting science is happening. So I want to thank you all for that. So as a biologist, the question I'm addressing is really how does animal generate behaviors? More specifically, as Judy has introduced, we use C. elegans as a really anatomically reduced, really simplified model to understand how animal behavior is specified. Yes. So an interesting thing about the animal behavior, specifically motor behavior, is really every single animal has a signature move. As human, we do walking most of the time. And you can also see some animals just like hoppings, and fish swims, and Drosophila flies, and frog crawl or swim. But despite of all this completely different outlook of the behaviors, there are some commonalities shared by the motor system that generate those behaviors, or there's some fundamental principles that can be shared by the way that movement has been generated. One specific thing, I'll try to generalize it as much as I can. So one specific thing is all the motor behaviors are rhythmic. So you may not realize this, but when you're walking or taking a breath or chew on something, this is something that you do rhythmically without even putting an input, without even thinking about it. Once it starts, you can continue to go. So this is a self-sustainable rhythmicity that's associated with all the motor behaviors. And second of all, to generate a really the motor behaviors are coordinated. So for example, if you look at this really famous early studies of looking at the walking of a cat, and they have electrodes stuck to the hind limb of the cat leg, and then recording specific group of muscles, electric signals. And you can see corresponding to the specific aspects of behavior, stands or swings, you have different group of muscles generating bursting activity patterns that correlates with the contraction of the muscle specified for specific behavior phases. And what's been notified immediately is the extensors and flexors, two group of muscles. They're firing completely off phase. And if you eliminate this phase contrast, these animals will generate constitutively condensed muscles and they will not be able to walk. So rhythmicity, and then also the off phase, the phase shift, or generating the antagonism of specific, making sure not just the muscle can contract, but the contract the right time in a specific pattern, a specific order is particularly important. And these are the things that all animals and all motor systems require to possess to be able to generate a coordinated behavior. So what are the cellular origins for those specific behaviors? This is one specific concept. The origin of the rhythmicity of the motor behaviors resides from something called a CPG, or central pattern generators. This is a very broad concept, but in simple words, it's a group of neurons which send out electric signals for the muscle cells to tell them when to contract and when to relax. Those are the group of neurons using the connections among themselves to self-sustained oscillating activity, so it can turn on and turn off and turn on and turn off in specific rhythm. And those central pattern generator neurons send out those oscillating electric activities to the specific group of neurons called the motor neurons. And it's believed that those motor neurons are the ones that using those patterns and instruct the muscles to contract or relax, and in specific orders. And that's the basis of generating specific behavior. But as you can imagine, if you generate a rhythmic chewing all the time and then you cannot stop, or if you're in the middle of walking and then suddenly there's a stone in front of you and you could not stop and reset that pattern, it will be really bad for you. So all this, even though this CPGs generated rhythmic behavior is self-sustainable, it can be stopped and retrained, reset the pace at any given time of the phase. And that depends on the stretch receptors or the so-called feedback loops. So the sensory systems send feedbacks to the CPG, even though they're not required to sustain the rhythmicity, but it can retrain it. So you can adapt to the environment and change the pace whenever you need it. Okay. So although conceptually this has been pretty well established for the more than last 20 or the last 2 decades, so 2 or 3 decades, for each individual animals has been an enormous challenge to really test some of the hypotheses and specifically to figure out how the connections of, first of all, what are the neurons that really generate the so-called central pattern generator? What are the group of cells that's self-sustainable and then can be entrained by the sensory feedback? And then specifically how the connections between those specific neurons is self-sufficient to generate that kind of ability. And then whether motor neurons or muscles or the stretch receptors plays additional roles than what the theoretical prediction has been implied in the previous studies. So for this, we have been studying the C. elegans for some very specific reasons. All the animals has a huge amount of neurons and then has magnitudes higher number of synapses, the connections of those neurons. C. elegans is probably one of the simplest animals in terms of number because it has only 302 neurons, and that's a very, very small number compared to any animal you can think about. This is like magnitudes of orders of smaller. So we and others naively thought that having a small number of neurons will make things a little bit easier. But it's really not. The animal compensates for the reduction of numbers by complexity of individual neurons function. Now we learn that. Biology is really smart, actually. But one important thing that I want to point out, because that'll be important for the rest of my talk, is this 302 neuron C. elegans nervous system is really only in the adults. So all the animals starts from an egg, and then it hatches into the larvae, and then larva, and then it goes through 3 different molding stages to get into the first stage larva, the second stage larva, third stage larva, fourth stage, and then into the adults. While the animal continues to grow in size, which is pretty obvious in this diagram over here, an important thing is the nervous system also continues to grow. So the so-called mature nervous system of 302 neurons, and John White, his colleague, has shown to be roughly connected by about 10,000 synapses, is really only occurring at the very last stage of his adult lifehood. When the animal was first born, it only has 202 neurons, and that's about a quarter of the neurons was missing from the adult nervous system. And specifically for the motor circuit, this is quite significant because it has only 22 of the 85 total number of the motor neurons. And the majority of the motor neurons was in fact added at the beginning of the second level stage, and then a few other neurons was added at the third level stage. And those neurons are pretty much contributing mostly for the reproduction, like coinciding with the maturation of the sex muscles. But a lot of these neurons are essential neurons that innervates a very, entire ventral side of the adult nervous system, adult animals. So the important thing is, this is not something specific for C. elegans, but it's becoming more apparent that all animals, including humans, continue to generate new neurons, and those new neurons continue to incorporate themselves into the existing nervous system without disrupting the functionality of the animal, including us, although teenagers tend to get a little bit awkward. So the important thing for worm is, although it lacks majority of the motor neurons when it was first born, but it crawls, and it always crawls. So this is the newly hatched L1 animal, and we contrasted the animals. So they're not black, they're really transparent, like what Judy said. But for you to see a transparent animal is a little bit difficult, so we put it into a black contrast. And this is an adult, close to adult. So the size increased by 10-fold, but the movement signature stays. And the movement of these animals is called sinusoidal movement, or what we call it as undulators. What signifies an undulator was really it's generating the body bends that in coordination of ventral and dorsal coordination. So this is the head of the worm, this is the tail of the worm, and there's a groove that basically travels from head to tail, and that coincides with the animal's forward locomotion. And what constitutes the groove is the contraction of the muscle on the ventral side that always coincides with the relaxation of the muscle on the dorsal side. And this traveling wave alternates throughout. And as you can see in the worm's world, at least in the laboratory's worm's world, it's been there for generations, that they like to go forward. It always travels the body bends from forward to backward, so the animals go, and then briefly it goes backwards a little bit. So the sequential order of the body bends contraction, the traveling reverses from tail to head. So it has an inherent bias to go explore the environment, and then but occasionally will back a little bit, and then resume the forward locomotion. So the question that we're trying to address, along with many other scientists in the world, is how does the worm really crawl? How does it utilizing the 202 or 302 neuron systems to really generate such a signature, stable, and also adjustable behavior? So as a scientist, you always try to break a big question into small questions. So for me, for us, or me and my group, we break this down question into 3 smaller questions. We break down the worm's locomotion into 3 steps. So for it to generate a movement, first it has the ability to generate the sequential body bends. And so you need to have a mechanism in place to initiate that bending and then to make sure the propagation of the bending goes smoothly and also in a directional fashion. And then this type of behavior constitutes 2 modes. Either is forward and backward, so there's sequential orders. And that forms the basis of 2 specific modes of locomotion. And then the second step is once you have that 2 bases, the animal needs to be able to switch directions. So it has to have something in place to generate the decision to make that switch. And then there's execution, the specific sequence of the steps to execute the changes of this reversal of the body bend's direction. And then the modulation is incredibly important, as I just specified at the beginning of the talk, because that gives the animal the power to adapt to any environment and then the change in the environment based on the sensory feedbacks. So if you put a simplified word, we're trying to figure out from those neurons which specific group of neurons and then the connections between those neurons can do each of those steps. Okay, so this is specifically how we're going to do it. And then specifically because these animals are continuing to change the composition of the neuron basis, so a bigger question is really how did they manage to generate such a stable behavior even though the cellular basis was changing? So to me, this is going to be a lifetime question for a lot of people. So I wouldn't be able to finish all this in the year for sure. But if I can identify a few of those components, I'll be happy enough. And I think today I'll just give you some of unpublished data that probably give provide some clues to some of those questions. So this is what the so-called wiring diagram of the worm. The worm is highwired, like all of us. We're really wired inside us. But I just illustrate this a little bit more to give you some ideas. So the highwired worm generate behaviors using neurons, and these neurons are dissected only here. And it has 2 parts, the soma of the neurons. This is where the cell body is. And then there's extension of this from the soma called the processes, and that's basically all those wires. What those processes allows neurons to do is to make contact with each other. And then where they make contacts, they can select to have specific subcellular structures to develop to allow these 2 neurons to talk to each other. Basically, one neuron can instruct the other neuron to become excited or de-excited, so that you can basically imagine the electric signals flowing from one cell to the other through those synapses. And then so you can have information from outside and then interpret by the brain of the worm inside and then through those wires and translate into motions of the muscles and then allow the animal to generate specific behaviors. In 1978, John White and his colleague did a manual EM reconstruction of all these neurons and then the connections, and then identify specific connections in the adult worms. So that's when they decided that the worms has 302 neurons that's connected about 10,000 synapses. And theoretically, combinations of those synaptic activation of those 10 synapses is sufficient to allow the worm to generate any behaviors that you observe in the lab. But finding that combination has been a very big challenge. And one thing I've told you, the worms do not always generate in the same set of neurons to generate those behaviors, so it's incredibly important to figure out what the connection looks like when the animal was first born and then was later continued to develop. So you might, but then that EM reconstruction was really done by two worms, and then only in the adults. So you might be wondering why wouldn't he does the rest of them, just because that one single two worms was taking him more than 10 years to finish, and then that's because it's a manual reconstruction and it's incredibly labor intensive and boring and very hard. So a few years ago, Jeff Lickerman's group and then R.V. Samuel's group in Center for Brain Science in Harvard University has decided to tackle this problem by automating this whole process. And essentially what they did, what Jeff Lickerman's group has done, is to develop a machine called Atom, automated sectioning of the worm. So you basically have the worm, turn them into a plastic, put it in this block, and start to cut them at ultra-thin sections continuously from head to tail. And each one of those tapes contain a little tiny speck on it, and that's the cross-section of the worm. And then if you use electron microscopy to scan it through, you will be able to see individual processes, the cross-section of the individual process of individual neurons. They form bundles. And then you can study the synaptic connections of these neurons so you can face them. So this is where we started it before I came to Radcliffe. So now you're actually zooming from the nose of the worm going to the mid of the body. All these big dark spots are the somas of the neuron, and then those specific fainter parts are the processes of the neuron. And this is a collaboration pretty much done by two people, Richard Shalak in Jeff Lickerman's group and Valeria Laskova, a graduate student in my lab in Toronto. This took quite a while, and then because there were a lot of technical issues to get through, as you can see, there are some cracks and then some dirts and all the things. And then some bad sections, mostly good sections. But then the real challenge came when we tried to so-called do the segmentation. The idea is you want to link individual object from each section with the next, so you know which axon, which processes connect to which neuron, and then so you can know where the connection happens between these two neurons. So that took more than a year of work by manually filling the spots of individual neurons, and then try to connect them with only about 500 sections. And this is the work done mostly by David Curson, undergraduate student in the Samuel group, and using a program developed by Daniel Berger, postdoc in Jeff Lickerman's group, and then Valeria Laskova in my lab. And this is really pretty pictures, and thank you, Daniel, for finishing that this morning. So this is the reconstruction of the first 500 sections in September 2013, and this is where that's basically from there, and then from here to here was the work since I got here. So Radcliffe, as you can see, you've done quite a bit of work. Well, sorry. So these are... But the sad thing with this is, even though this took so long and there's so much labor-intensive work from highly intelligent and hardworking people, these are really just a quarter of the first-born RV. And so far we have about 3,000 or 4,000 sections, and we know it will take at least like twice, or at least like 3 times more in those sections to get to the end of the worm. So this may not be the really ideal way to go unless we have unlimited fundings and times. So one thing we thought about doing is, because the worm was very long and then it's really thin, so instead of doing a section this way, if we do a section this way, then we would be able to get by with only a few hundred sections instead of several thousand sections. And that turned out to be a really good way. So this is so we finished reconstruction of so-called longitudinal reconstruction of the first L1. This again involves a huge amount of people, because it turns out even though the section has much smaller number, but they're so long you have to stitch them together. And that takes the computer power and also some manual stitching and cutting of papers, which they have over there. By the way, you can go take a look. So a lot of work. But we finished that, And then we now finished 2 or 3 sections of the worms, and then we're continuing the SEM reconstruction of the other 3 animals. And what this would tell you is, oh, do I have enough time? Yeah. Yes. Okay. So this is just a close-up of what you expect to see. So these are individual neurons, and now we're doing the segmentation of those individual axons and linking each of them back to the neurons. And then more specifically, we got a synaptic resolution. So this is the so-called synapses of from one neuron is talking to the other neuron, because this is exactly where the release of the signals from this neuron comes into here, and meanwhile this neuron will talk back to this neuron, because this is where the talking center happens. So at this resolution, we hope to try to finish reconstructing the firstborn young adult, young larvae stage within the end of the year, and then we can really compare the connectivity of this juvenile L1 to the published adult data. But they've already talked from other very famous scientists in the US and other groups that they would like to consider reconstruct throughout all the larval stages as well as the adult stage animals, so that we can have a number, not just one, but multiple ones, so we can get statistic significance and to get a real sense about stereotypicity of the individual animals. Okay, so meanwhile, while this work is going, there are other things we could do to really try to understand how the connections might actually generate behavior. The reason is, if you're trying to figure out the connections of the individual neurons, well, you're still trying to figure out how which neuron connects to which. There are other things you could do by, for example, if you take out a specific group of neurons and then see what the effect is on the behavior, you can already establish some kind of fundamental functional basis for what they might be doing. And then if you have that, then you can relate to what the synaptic connection between the neuronal connection could be doing. So again, this is the work that the so-called functional annotation of the worm's nervous system, again, is done in collaboration with the Toronto group, postdocs, Taizo, Shangban, in my lab, and the graduate student Asuka Guang, and then a postdoc fellow Chen Wan in the Samuel lab, who's actually going to take off, set up his own group in China in the fall. And Daniel, of course, the research partnerships from Radcliffe. So I'll just give you a brief outline. It will be very quick. I'll try to make it as simple as possible. So if you want to study the motor behaviors, we know that the motor behavior is ultimately driven by the muscle activity. So if you can figure out what neuron specifically innervates those muscles, then this is the first order, the lowest order of the nervous system, called the motor neurons. And then if you know the connectivity from the other motor neurons, then all the input, all the neurons that instructs the motor neurons called the premotor interneurons. And then those premotor interneurons inputs neurons called the other interneurons. And then ultimately, those interneurons receive synaptic inputs from the sensory neurons. This is how the animals receive sensory inputs, activate sensory neurons, activate interneurons. Interneurons have the information or decision made to the premotor interneurons, and premotor interneurons give their orders to motor neurons, and motor neurons tells the muscle what to do. So we can specifically, what we think is, because we know where the muscles are, we know where the motor neurons are, we know where the premotor interneurons are, we can start it to gradually, stepwise, systematically take out those neurons, for example, the motor neurons, specific premotor interneurons, and see how it does affect the behavior. And this has been easier, it's easier to say to be done, because how can you go into an animal to kill specific cells? This has been, so it makes it much easier now that we can use a so-called optogenetic approach. So this is a protein called a mini-SOC. It was invented in Roger Shen's group at UC San Diego about 2 years ago. Essentially, this is a protein that can be activated by specific spectrum of light, and then upon the light stimulation, this protein will generate free oxidate. And that will generate oxidative stress, and the oxidative stress is bad for your cell. And it's so powerful if you actually pop this in the specific mitochondria in your cell that you can basically generate enough free oxidative stress that the cell can die within a few minutes. And what's really powerful about this approach is completely light controllable. So you can have the animal expressing this toxic protein in the cell and grow up totally normally, as long as you avoid the specific spectrum of light, in this case the blue light. And then any stage you wanted to see the neuronal functions, then you can just shine them under the specific light for a few minutes, and then you can immediately see the shift in then those neurons because of the specific neuronal depth. So this is a specific group of the premotor interneurons kill. I will get back to. So this is the first approach. And the second optogenetic approach to understand the nervous system function to contribution to specific behaviors, to have a way to monitor the activity of individual neuronal groups in the moving live animals. And this is also technically extremely challenging things to do because all the previous methods to detect the neuronal activity is to really stick an electrode inside the animal to monitor how this membrane of the neurons change, the membrane voltage change, because when the neuron fires, the voltage will become depolarized. So, but then if you stuck electrode in it, you can see the animal will not be able to behave normally. And then there's a lot of other parts of the animal is so deep, it's almost impossible to get precise access to those specific neurons. So again, Roger Shen's group, and then a few other groups. Oh, this is not Roger Shen's group, another group. I then generated another genetic tools to allow us to monitor the neuronal activity without sticking anything into it. This is one thing is so-called a calcium sensor. So this is taking advantage of the property of the neurons. When the neuron becomes excited, it's going to release calcium, the specific iron inside the cell. So if you can monitor the calcium concentration in the cell, you can predict whether this neuron is firing or is not firing. And then if you want to indicate the calcium concentration, what they did is they took a protein called the GFP. So this protein will fluoresce under specific light that's basically taken from the jellyfish. They cut them open and then flip them aside. So this essentially makes a non-functional protein. So it no longer fluoresces. But then it stuck specific modules at the two ends. These are the proteins that specifically binds to calcium. And when they bind to calcium, there's a secondary tertiary structure change and causes the entire protein to refold. And this refolding of the protein basically allows this non-functional GFP protein become functional again. So without calcium, GFP has very, very low fluorescence because it's basically protein cut into half and then put in a reverse orientation. But upon the binding of calcium, that means if the cell is having a lot of calcium, now this protein will reconstitute and give a high fluorescent intensity. So just by monitoring whether the fluorescent intensity was increasing or decreasing, you know the calcium is increasing or decreasing, and then you know whether the neuron is active or inactive. A similar idea was a voltage sensor. This is something directly using fluorescent intensity change to indicate whether the membrane voltage was decreasing or increasing. So this is the work of Daniel Wall, and this is something we just started last year, and then we haven't been able to follow up. It's a very new technology. But the data I'm going to show you today, so this is so-called free calcium imaging. So this is a worm that's generated by Chen Wang and Samuel group. So he's basically expressing this calcium sensor in the specific sensory neurons, or AIY. And then he's targeting this fluorescent intensity and tracking these fluorescent dots while the animal is moving. And then this is a color coding of the fluorescent intensity. The cooler it is, the lower activity it is, and the hot it is, the higher activity it is. So as you can see, when the animal is moving away from a gradient, it's cool. And then when it's changing direction, moving towards a specific chemical gradient, it becomes hotter. So that means this neuron is really being activated when it's migrating towards specific gradient. And all this is being done when the animal is completely intact and behaving normally. So combining these two techniques, I'll just give you some highlights of our unpublished data in the motor circuit. So using this mini-SOC light-activated ablation technique, we ablated specific groups of motor neurons and premotor interneurons, some of the interneurons. And then one striking data we found is if you ablate all the so-called premotor interneurons, these are the neurons that sends all the input to all the motor neurons. So now all the motor neurons has no inputs. It's completely isolated from the rest of the nervous system. You got a very unhappy worm. So this is without light and it's not moving. And when anybody look at it, say, oh yeah, of course, because there's no instructions from the upper layer system, so they do not know how to move, so they're not moving. And so this muscle is just not doing anything. but this is not what you're seeing. So this is using so-called calcium imaging. So we're trying to monitor the specific muscle activity. If you think the worm is not moving because the muscle is not doing anything, and this is not exactly what you observe. These animals has very, very high activities in the muscle cells, and it's actually generating the local bends. The only problem is those bends is not traveling. So this one, for example, this bend is constitutively dorsally bent, and this is constitutively ventrally bent and constitutively dorsal bent. And this is in contrast to the case of the wild-type animals. When it's moving towards certain directions, you can see a traveling calcium wave along the body. So essentially, when you kill the premotor interneurons, you're not resulting in an animal that's completely paralyzed, but you're constructing a worm that's completely frustrated. It's a physical term, in fact, is because it has a constitutive activation that do not travel. Essentially, that's the point. So there are two classes of motor neurons that generate the body bends, the A class and the B class. These neurons have very similar autonomy, but except the axon, so the process's orientation is opposite. So it has always been hypothesized that these two classes of neurons might be responsible for forward and backward locomotion. So because these animals, when they do not have the premotor interneurons inputs to those motor neurons, they generate a standing calcium wave. So we're trying to figure out whether this calcium wave is because one class of motor neurons become constitutively activated in the absence of the input, or in the absence of the input, both classes of the motor neurons that's supposed to be generating only two directional movements are simultaneously activated. So you're having a calcium wave that's standing in the middle and do not distinguish directionality. So we can take that trick by, again, ablating all the premotor interneurons, has both A and B class motor neurons completely suspended from the rest of the sensory input and interneuron instructions. But then on top of that, we can kill individually the B class motor neurons. And as you can see, now these animals, like from there, is generating backward movements. But occasionally, when the tail of the worm is generating backward movements, the head is generating forward movements, so generating a pulling on the side. So have a very flat end. And then you can kill all the premotor interneurons and the A-class motor neurons. You do not see those kind of constitutive activations anymore. This animal is generating a very slow forward movement, dragged by the head swing, and then it doesn't back. So what that essentially tells you is, in the absence of premotor interneuron inputs, these two classes of motor neurons become antagonizing forces to each other. And then the A and B class motor neurons become simultaneously activated at the same time and the same force, so the animal cannot distinguish whether it's going to go forward and backward. And in fact, without the B class motor neurons, A motor neurons themselves is capable of generating a sequential activation that can generate backward movements. So this is a more electrophysiology term. So that kind of behavior of a neuron really qualifies them to be called the CPG quality. That means this neurons without any inputs is able to sustain a sequential activation, and then including the activation, the oscillating activation and inactivation activity. And if we can detect that this is all by behavior and also some of the calcium imaging analysis. But the more direct way is to figure out whether the membrane potential changes oscillates spontaneously in those background. Shambang Gao postdoc in my lab developed a C. elegans prep, sticking the electrode in the dissected worm prep, cut the head, cut the tail, cut all the premotor interneurons, cut the B-class motor neurons. So this is only the signal from the A-class motor neuron without any sensory input. You see these oscillating electric patterns. So we have some specific models over there. We believe the C. elegans locomotion is driven by multiple CPGs. Backward CPGs is actually coming from the motor neuron themselves, called A-class motor neurons. If you don't have any inputs from the upper layer input neurons, this A motor neuron CPG is constitutively active, and then it can actually inhibit the traveling of the wave by activating the B-class motor neuron that typically promotes the forward locomotion. And then when you have these two neurons classes activate at the same time, without any modulation to separate them, this will prevent the traveling of the neuronal activity that drives the backward locomotion. So this actually goes very nicely with the previous work we published that Taizo Kawano in the lab has previously identified the premotor interneurons can be divided into two groups. One group of premotor interneurons is actually using a synaptic connection to inhibit the spontaneous activity of the A-class motor neurons. And then the other class of premotor interneurons activate the B-class motor neurons. And these two classes of motor neurons have this off-phase high-low activity pattern, and that instructs A and B motor neurons' basic neuronal activity to be separated. And so in wild-type cases, there's two things. A-class motor neurons was spontaneously biased towards the low activity state, and the B-class motor neurons biased towards high activity state, and then they're always off sync, so that you can always have the animal generate specific directions. So I think this is really as scientific as we're going to get over there. And I am really excited that we're going to be able to now try to put together to figure out the synaptic connections between premotor interneurons and actually find the synaptic connections that's responsible for the specific act between the A and B class neurons communication, the premotor interneuron and motor neurons communication. And then I think the collaboration will continue despite the ending of the Radcliffe. But me being here has really been helpful, has been really instrumental to push the project forward, and we know exactly what the next phase would be. So the last slide is this. Because I want to save the trouble of whenever I give this talk in the scientific meetings, the question is, how do you think your research is going to benefit human health? And I always have those generic answers to a scientific audience. Understanding the basic biology is always instrumental to understand that. But deep in my heart, I would want to say, personally, this is just my passion. I like it. So it doesn't matter. I like movements, and I like to understand how movements is generated. It's impossible for us to understand how we generate movements nowadays. We just don't have the tool. It's too complicated. Personally, I do believe what Martha Graham has said is true. Dance is a hidden language of the soul, and the motor behaviors is really an unwritten expression of the soul when you do not have the words adequately to express your feelings. And I believe all the artists will say that not dance is the hidden language of soul, maybe painting or the novel or writing is the hidden language of the soul. And I think for me, the work that I do is the hidden language of my soul. So with that, I take a quote from another scientist. He's basically saying there are multiple CPGs responsible for different motor behaviors in human. Breathing is controlled by a specific set of CPG from our walking CPG, and the swallowing is been by a specific part of CPG, and it's very important that we should study it. And the last slides, I want to thank everybody. So the incredible staff from Radcliffe. It's been a really amazing year, and thank you all for taking such good care of me. My research partner, Danielle Walls and DJ Brooks was there, and members of my collaborators, Avi Samuel and Jeff Lickerman, for making it such a scientifically rewarding year. And Nali and Catherine are my dance instructors in the art center. It's been really nice. I really thank them for what they've done for me, and all my Radcliffe fellows and friends for being here. Thank you.